All right, so standard reminders. So as with any other week, another muddiest point to do this Sunday. There's another concept check for the bottom half of this unit due Sunday, and then a week from Sunday, so next Sunday, there'll be the first mini project. This is the only mini project that is not dropped. So this is a project where I'm going to ask you to optimize a very simple numerical function that has a couple of to minimize the function that has a couple of different local minima using a genetic algorithm that you build from scratch. And so we're going to start talking today about the genetic algorithm and how you build that and what a basic genetic algorithm looks like. I tried to provide a bunch of feedback on muddiest points from last week, and I added some additional notes documents for those who are wondering about the mathematical implications of the drift barrier, and just generally wanted some more details about that diagram I had at the end of last lecture. I thought a motivating example, I was at a new bakery, Paris Baguette, with my wife over the weekend, and I was sitting there in the bakery, and it happened to be the two owners were there talking to somebody from corporate who was stopping by and giving them feedback or whatever, and there was a sort of the manager was walking around the store doing a lot of different things, doing them very efficiently. And then there was this, I'm assuming a high school kid behind the counter who might have been a new employee, but was doing absolutely everything wrong, like a real, pimple on the butt of progress, and looking at that, I looked at these two employees and I was thinking, if you're trying to find like the best employee, then this would be a great example of a very high selective pressure environment, because you had this manager who's very good at her job juxtaposed next to this paperweight who was just getting in the way. And so and you had the owners and somebody from corporate sitting there evaluating all of these different things. And so you might think that the next day you would continue to see the manager there, but you'd see somebody else completely different behind the counter. And that's what a high selective pressure environment will do, is that very quickly you see a turnover of the environment. It looks very different than those that were high fitness are definitely there to stay. Those that are even slightly lower fitness are purged from the environment. But that's not what's going to happen, right? So that kid behind the counter they're probably going to give him a break, going to keep training him, and eventually he might get better. So that, when we talk about reducing selective pressure and the mechanisms for that, that's kind of a motivating example for that, is to say that just because the first candidate solution comes in, does things a little different and gets very low fitness, doesn't mean you immediately throw them out. You might let them stick around. And so we're going to talk about fitness scaling today and functions that do that. And the function of fitness scaling is to say that although it's clear that one solution is far more fit, is a better suited than another, we are going to try to preserve diversity, because having diversity in terms of our solution space gives us the ability to explore new solutions. And it might be that the other solution that is poor fitness will eventually change in ways that differentiate itself from the other high fitness solution, but allow it to achieve high fitness in a different way, and maybe even achieve higher fitness. So sometimes you have to tolerate lower fitness for short periods of time to allow for exploration to find higher fitness. And that's the reason why we're not going to want super, super high selective pressure algorithms. And then likewise, when we finally get into multi-objective, we're going to learn a way to use a multi-objective algorithm to create a better single objective algorithm. It's called age fitness Pareto optimization. We don't have to worry too much about it, but the key point there is that they age structure the population. And they basically say that if you are a recently arrived candidate solution, we are going to not require you to have as high of a fitness as older solutions that have been around for several generations. And that's exactly what you see in that bakery there, is you give the newer employees a pass because they're still learning, they're still evolving. And so but if that newer employee, after several weeks of working there, doesn't improve, then at that point they stop getting a pass. And that's the exact same thing that happens with age, fitness, Pareto optimization, which we'll talk about these things in Unit 3, where in that case, you keep track of the age of these solutions, and they can take a hit on fitness early, but once they all start getting to a particular age, then they have to start competing in terms of their fitness or in terms of their optimization objective, and then you might start clearing them out. So you might have high selective pressure within an age cohort, but you might have low selective pressure across the population. And that helps maintain diversity in your solution set, which allows for exploration, but then around any particular solution allows you to fine-tune it exploitation. So those are the types of things I want you to be thinking about as we get into these concrete implementations of these algorithms, is that we're going to see these hyperparameters and try to think, how does this hyperparameter affect selective pressure? How does this hyperparameter affect the diversity of solutions, and so on and so forth. So that's kind of the lens that I want you to have. All right, questions, yeah?
How would you, if you have something like f of x equals 1 over x, the graph looks like that, and there's a negative infinity for the fitness function. How would you be able to like explore to the, if you start at the right side, how will you explore to the left side, because that's just going up and up?
Right, well, so that's the, and so, I mean, this is an issue with any optimization function, but if you have, if you've got x, you've got f of x, and you do have something that has an asymptote like that, fast enough. So if you have something that has an asymptote like that, if the way you're searching is just doing incremental updates, then you can't get into that trap. Or if you're trying to maximize, or if you're trying to minimize, you might be over here, and minimization actually takes you in the wrong direction of solutions that are even lower. So part of the advantage of this, reason why I talk about diversity preservation, is that hopefully you instantiate a diverse population and you maintain that diversity. Because then that allows, so if there are these weird barriers, you've got some individuals on the other side of the barrier. And so you've got individuals that are searching in very different parts of the X space. But then the proliferation, how the survival of those individuals depends on their relative fitness to each other. So they can explore in very different places, and then they come back at the end of the day and say, how well did you guys do? And then based on that performance review at the end of the generation, then you decide, you say, okay, well, you three can continue to move, you fourth I'm kicking out, and I'm going to bring in someone new who looks like these three. And then that helps deal with that. So, but it might be that you just haven't jumped over one of these gaps. And so, that's part of the reason why we have mutation, and part of the reason why we're going to see as a hyperparameter is mutation rate. That's the amount of spontaneous deviation from the existing population that you're just constantly trickling in. And that might allow you to populate a region that is walled off from all the other regions, but you can kind of tunnel through it with mutation. So I think all that will become clearer as we start seeing more of these algorithms and how they work. All right, any other questions about that idea of selective pressure or just anything so far? Assignments coming up. I think a lot of the questions that came up in the muddiest points, as we start seeing these concrete implementations, we'll start to resolve them. So let me adjust my eraser settings here. All right, so as a reminder, basic engineering design optimization problem, minimize some function, some scalar function, where our design space has been defined, so there's a feasible set. And so we are going to use a genetic algorithm to approach this problem. So how do we start mapping some of these biological terms into the numerics that are in our algorithm? Well, our decision vector, X, that is our chromosome or our genome. So whereas in biology, genomes are made of multiple chromosomes, at this point, there's no leading genetic algorithm that has shown a strong advantage of creating multiple chromosomes. So you'll just see those two terms. Generally, people use the word chromosome, but you might see the word genome, and they're basically synonymous in the context of a genetic algorithm. So it describes where each decision variable is located in the vector. You know, it basically encodes the set of decision variables as a string. So the order here matters. That's going to be a key thing about genetic algorithms versus other evolutionary algorithms. This encoding, this idea that you've got like 100 variables or 1 billion variables, so it could be 1 billion weights in a neural network. It could be 100 ways that your design of a chemical plant could differ, and so on and so forth. And you are loading them up into this vector. And this vector could have lots of different data types in it. It doesn't have to all be numbers. If you do want to encode your vector in a MATLAB matrix where every row is one of these, then it does help that they're all floating point numbers, but they don't have to be. But the string-like characteristic, the fact that it's a sequence, the order matters. It has a linear ordering. That will become important for the action, primarily the action of recombination or sex. So we will get to a recombination operator where two candidate solutions can come together and have offspring that share features of both. And that requires them to line up their sequences next to each other and choose points that below which they'll swap and above which they'll keep. That's something we'll call crossover. And that crossover is set up by everything being located as this string. And so that is one key differentiator here. So examples of that, like I might have designing a car, so I might have one variable, my vector, x1 might be color, x2 might be wheel diameter, x3 might be my fuel type, but I might do a very different encoding where I might, even this color might be broken up into its binary bits, its binary digits, its bits. So I don't have to, each one of these little vectors here doesn't have to be a real number. It could itself be some other encoding of a portion of a number. And you might seem like, well, why would I want to do that? And you might find that it provides more interesting ways to move through the search space. Because once you separate these things up, then you can mutate each digit, you can mutate each bit. And then when you bring, and we'll see how recombination operators work, but when you bring two candidate solutions together, they can swap some of their bits and not other bits. So Gesundheit, we were going to find that recombination has like an interpolation effect. So if I've got a solution here that is, it's a solution that's got one temperature of my chemical plant, and this guy's got another temperature of my chemical plant, then it turns out that the way recombination works, your offspring might end up being a kind of in between the two of them. And that enforcing that in-betweenness through this recombination sometimes is easier to do when you break that one number up into multiple digits and then make those explicit, make that explicitly part of your chromosome. And they don't have to be binary, they can also be decimal. So there we go. So this is going to be like your mini project one. You could have a decimal, so you could have like this being, one, the number 0. If I had like, 0.1356, this might be x1, this might be x2, this might be x3, and this might be x4. And my convention is that each place, each gene in my chromosome is a place value of this number. And again, that might seem a little silly. Why don't I just have a real valued x1 that is the whole number there? And I think, I'm hoping as you play with mini project one, you'll see that having these encodings this way can maybe allow for more interesting ways to search the space. So if I just had this as X1 and I wanted to manipulate it, mutate it, explore, I might just have to add noise to that single number. So it might be that this is my individual, and if I want to try another individual, my only option is to add some sort of noise to this individual. But once I break this individual up into this string, then now I can mutate each digit separately. I can add noise to each digit separately. And then if I bring two individuals together, I can have them swap their digits, and we can talk about what are the numerical properties of them when they swap their digits. This ends up becoming, modeling alternative ways to search through the space. So that we have a lot of flexibility in how we choose the encoding of this string. And, again, this is only 4 variables, 4 genes. I might then have another 4 for another number, another 16 for another, and so on and so forth. So this long string could have thousands of members, where then there's sections of them, like these 10 represent this decision variable, this 1 represents this decision variable, this 1 represents this decision variable, and so on. So all organized as a string. So that's what we mean by a chromosome, where each spot in that chromosome, each variable in the vector is our gene. Questions about that? Yeah.
I have a question on that. So here it's written that the set of decision variables becomes a string. So can we say that this string that was a particular set of genes is a allele?
Alleles will be on the next page.
Okay.
Yeah. So any questions about this? We're going to get to alleles, which are so this is the layout. The alleles will be the actual values that you draw. So I will say that this here is a genotype, and each spot along here is an allele. So this individual carries the 1 allele at position 1, the 3 allele at position 2, 5 and 3, 6 and 4. And this individual's genotype is 1, 3, 5, 6. That would be how we would phrase that. Now, what is the value of 1, 3, 5, 6? Is this good or is it bad? We're gonna need a notion of a character, which will be our optimization objective. And the value of that character will be its trait value. And that's all coming up here on the next slide. Other questions? All right, so let's get to that. So for each gene, we define a set of alleles. So that might be this continuous range here. So it might be that for the X gene, it can take values in this entire continuous range from 45 to 206, every real number in between. It's totally fine. These are all alleles. These are all variations, values that could fill in here. Alternatively, we could have discrete sets. So we could say that, and these all could be within the same chromosome, that X1 is continuous, X2 is discrete and binary, and X3 might have 10 values. So these are all our set of alleles, and these are all the genes that are the locations where those alleles are found. So biology, generally speaking, we only have discrete sets of alleles because of how our DNA is a discrete molecule, discrete information molecule. But we have the ability to store floating point numbers in our data structures pretty efficiently or pretty effectively. So we also, unlike biology, can have continuous valued alleles, which is a little weird if you talk to a biologist, but it's something we can do because our data structures are different than DNA. Okay, so individuals in a population vary in their combinations of these alleles. And each individual combination is a genotype. So we can have individual 1 eventually here. So we can have individual 1 that is going to, this is going to say X1 vector here. And I don't know why it's slow today. This room is, so individual 1, it's a vector with these three variables. Each variable is the gene. the particular gene variants that this individual has are listed right here. So individual 1 has allele 53.3 at location 1, has allele 1 at location 2, and allele 8 at location 3. I'm going to use the notation that if individuals are listed as vectors, then the first subscript will be an index of the individual. And then if I look at the variables, then the second subscript will be the index of the gene. So that'll be the location in the vector. So Xij, I is the individual, J is the gene. And then the particular value at Xij is the allele at that value, and all of those values together are the genotype. So this is one genotype in the population. Now there could be another individual in the population, X2, which should come up here in a second, and X2 could have a completely different set of alleles at some positions. So 19 pi, I just put in there for kicks, 19 pi happens to be in that range, 45 to 206. Something around 60 or so. And then they differ at the second location, but they do not differ at the third location. So you're going to have some alleles that are shared and some alleles that aren't. And that works well for us because as we search through the design space, we're going to find that certain decision variables are going to be like, this is a good set of these decision variables, and they become decoupled. They might be decoupled from the function of these variables. And so you might end up optimizing one set or one portion of your chromosome while you're fine-tuning, trying to figure out the right combination of the other portions of your chromosome. So for whatever reason, these two individuals agree in some places and disagree in others. And so we are maintaining a population of these genotypes, and we are trying to keep the ones that are doing well and then slightly perturb off of them to explore and then throw away the ones that are doing poorly. And if we have ones that are doing well, then this recombination, which we're going to be introducing, that recombination may take two that are doing well and end up creating offspring that look very different from both as an attempt to try finding new combinations. Because one of the things about these things is if you don't recombine, you can get things kind of correlated with each other. Like in order to get a 1, it's maybe always with this 53.3, because for some reason the 1 hasn't come up in the mutation or whatever, but it seems to be a pretty good value. But that 1 is always with this 53.3, and maybe that's a bad value. So we might want to get this individual's 1 and another individual, try that out. So recombination, sex, between these two individuals provides a way in which parts of the genotype can get shared with others while masking off other parts of the genotype. So again, we'll get into that. For now, just so you know, genotype is the individual values. Questions about that? Okay. All right, so now how do we evaluate each one of these genotypes? Well, then we need characters. So remember, characters are observable dimensions that we may tie to performance. They may just be things that we can observe, but we actually don't care about, in which case they're neutral characters, but they may be important. So for example, for a given design of a car, I can map that to the car's top speed. Well, top speed might be important to my stakeholder. Top speed is something I could observe, but in a particular stakeholder, they may not care about top speed at all, in which case, just because I can observe it doesn't mean I want it to be in my optimization problem. So anything that you could observe, anything that you could assess, we're going to call a character. And when you evaluate the character for a particular genotype, then we're referring to that as the trait of that individual. So an individual can have lots of different traits. Can be evaluated along lots of different characters simultaneously. So we might have the top speed of the car. We might look at the same genotype and be able to evaluate the weight of that car. We might be able to evaluate the battery life of that car. if we're sticking with the car examples here, jumping around to a different example, if the X, if the genotype is actually the sequence of all the weights of a neural network, then we can say that neural network, what's the classification loss for that neural network? So if I'm trying to make a neural network that looks at a bunch of pixels on a screen and decides are those pixels a cat or a dog, and I have a bunch of example cats and dogs, then I can create a loss function that says for every cat, how close did you get to cat? For every dog, how close did you get to dog? Add up all that loss, and that's our classification loss, and we might want to minimize that. Now, at the same time, there might be, if those who've worked with neural networks know that sometimes we don't just care about the loss, the classification loss, but we have other things we tack on. And so in neural networks, there's something called a regularizer, which helps to say if there's lots of different ways that you can build a classifier. So you've got more weights than you need, more your genotype, your chromosome has more degrees of freedom than are needed for this problem. Then we would like you to choose the set of weights that solves this problem, but also is most likely to generalize. And the so-called regularizer helps to do that. So we can say we want to minimize the size, the norm of the weights that you pick. And we want to do that somehow simultaneously as we minimize our classification loss. So there can be these multiple objectives simultaneously. And so regardless of we haven't talked about how we're getting this into fitness, but right now I'm just saying that the way we map, we talk about traits in biology. What is a trait in a genetic algorithm? A trait in the genetic algorithm is the particular evaluation of different metrics that you can apply to that genotype, to that vector, that decision vector. So for a given genotype, you bundle all those trait values together, and that is the so-called phenotype of that individual. So that's just the set of different scores that individual has on things that you might care about. Now, you might not care about those things. The fitness function we're going to use to define what we care about. Question?
So is this a multi-objective optimization?
We can't. Well, that's when we get to fitness. In general, I'm defining this generally as multi-objective. We will then I'm going to specialize it, say for the GA that we worry about in this unit, it will be a single objective version. Then in unit 3, we'll come back and then show how we do it properly for multi-objective. But the framework I'm building here is I'm just going straight to the general case where we're defining multi-objective. We assume that there potentially could be multiple things you care about. And I'll show examples of fitness functions that start pulling these together in kind of trivial ways to show you, at least even with a single objective fit, or even, I don't want to get ahead of this. Okay, so that brings up fitness. So I have to define, fitness here. So in biology, I'll say this stall while I'm waiting for Zoom to catch up. In biology, fitness is a statistical descriptor of reproductive success. In biology, there is no fitness function. There's a fitness statistical representation. So in biology, I can look out at a group of individuals and I can get their genotypes. And then I can look at how many offspring those group have and get their genotypes. And I can watch how alleles in that set of genotypes changes over time. And I can then define fitness of those particular alleles by describing them statistically. So I don't know why this allele seems to be spreading through this population. There's some ecological reason why it's really good to have short white. So this allele happens to cause wings to get modified so they're shorter. And in this particular ecosystem, it's better to have shorter wings. So those with shorter wings have more offspring, and that allele tends to spread. And so you start seeing more of that allele, and with that, shorter wings over the whole population. So there is some complex system that is driving that allele to change. I'm not building a model of that. I'm not building a physical model of how you get from that allele to whatever biomechanics make that allele spread. I just look statistically out of the population and I say, hey, I just noticed that allele is spreading. And I can map, for the different alleles that are there, how many offspring per generation am I getting? And that's my fitness function, is a statistical description of what's happening in biology. In our case, it's going to be prescriptive. In our case, we are encoding our values. So we aren't actually allowing these things to reproduce and allow. So there are people in the artificial life community, like Charles Ofria up at Michigan State, who built simulators that actually have little virus-like code that interact with other code and try to reproduce. And he can then do what the biologists do. He can say what code snippets tend to make these little simulated organisms reproduce more and more. And he doesn't know those things ahead of time. He describes them statistically and then uses them to study what's special about those code snippets and so on and so forth. That's not what we do. We say to our stakeholders, what's important to you? What characters, what things can we measure about this genotype that are important to you? And then we try to encode those as a quality of a solution. If you can be better in this and better in this, then you should have a higher fitness than something that, all things being equal, is lower than those things. So fitness helps us encode how we want to sort genotypes by all the different ways that they can differ. And so it's prescriptive for us. We're not saying that biology has some fitness function that it's implementing. In biology, it's statistical. For us, it's probabilistic. So it determines the number of offspring that we are going to. In biology, it describes the number of offspring. In engineering, it determines the number of offspring. And higher is better. So there are certain, question.
How would you find the success of an allele if it was a continuous range?
What do you mean if it wasn't a continuous range?
If the X1 belongs to 45 to 206, how would you say that an allele is more successful? Would we be taking an average or something?
Well, that's if we wanted to do what the biologists do, then they use general linear mixed effects models to do what you're saying. So they look at all the different times, they look at all of that, like this person, how many times does this particular allele show up across these individuals? And then let's do statistical patterns across these individuals, effectively averaging across individuals that have that allele, compared to other individuals that don't have that allele, or that have other alleles. And then that will explain, there'll be a certain R² value that'll explain a certain amount of variance. And things that can't be explained by that variance have to do with other alleles or drift stuff, like things that don't correlate to phenotype and so on. But we don't care. I mean, we could do that, but it would just be kind of a moot point. Here, what we're saying is you take all of these alleles that are sitting inside these genotypes, and you then evaluate these characters. So you evaluate a loss function, for example, and a neural network. And you know that lower loss is better. So for this individual, it has lower loss than the other individuals. So we are going to allow this individual to persist and have more offspring than other individuals. So that means in the next generation, then there will be more individuals that have genotypes that look like that previous individual. So that will cause the alleles to spread, but that's because they're bundled in this package. It might not be that allele. That's why I brought up quantitative genetics, is that usually, like if you think of a neural network, it would be silly to say that this neural network that has 1 billion weights is successful because of the value of one particular weight in that 1 billion weights. It's the suite of those weights and how they work together that determines the loss of that neural network. And so it's true that there are certain weights that there might be a family of weights that end up being the ones that are selected by this process, by gradient descent, by whatever metaheuristic you're using to tune your neural network. But we view that as a performance of the individual level, not as a performance of the individual weight level. And if certain individuals have commonalities, then those commonalities will enforce regularity that spreads along with them. Does that make sense? Great. So what are the properties we need for fitness? So fitness is a scalar, even though we theoretically are considering multi-objectives here. Ultimately, at the end of the day, in biology, it's about how many kids you have, how many offspring you have. And that is the same thing in an evolutionary algorithm. It's about how many offspring do we generate that have this genotype or a genotype that is derived from it. So fitness has to be a scalar. It's not going to be a vector. So we have to have some way of taking these multiple characters and smashing them into one fitness value. The other thing is that for genetic algorithms, fitness has to be non-negative. Now, we're going to find that there's selection operators that we're going to implement, so that's going to be something we're going to talk about. And there's one particular selection operator, kind of the default selection operator, called fitness proportionate selection, which you don't have to write that down, we're going to get to it. That is the key one that requires fitness functions to be non-negative. Now, if you use other selection operators, you might be able to relax this. But that's sort of, again, if we want the analogy, like it doesn't make sense to have negative offspring. So this fitness should somehow be related to number of offspring. And so you wouldn't say you have -5 offspring and compare that to having 10 offspring. So we want it to be non-negative to sort of make sense and to allow us to use selection operators that are inspired by biological processes, sort of. And then the last thing, which I've kind of already said here, is that fitness, scalar fitness that is non-negative allows us to rank or sort genotypes by quality. So that's kind of the key point of fitness here, is it takes something like, if you differ in 4 different characters, 4 different dimensions, like this is your speed, this is your weight, this is how much you cost, and this is how much energy you use, or your battery life. You have these 4 different ways. It's easy to compare along each one of those dimensions, but how do we compare across all of those dimensions? Somehow biology does it. Somehow biology makes a determination of the fitness of an individual, even though that individual differs in all these different ways. Our stakeholders certainly do it somehow. So we have to somehow encode that somehow into our fitness function, even if we're measuring, because once you have more that are 2 or more characters, there's no way to sort them. You can sort the real numbers, you can't sort the complex numbers, right? Like once you have 2 dimensions, there's no ordering anymore. So we need to restore an ordering, and that is what the fitness function does. It restores an ordering to this complex character space. Questions about that? Okay. All right, so example fitness functions. So eventually here, so the first example that I'm going to show you here is, what if I want to minimize a single character? The character is the cost of a product, or the character is whatever I choose to minimize here. It could be a maximization problem, in which case the F would be a negative, and this would become M plus. But there's this capital M that I've put up here. And the idea here is that if I maximize F, big F, I will minimize little f. And so that's how this fitness function is built, so that maximizing the big one minimizes the little one. Now that's why I have a negative between there, M minus that. But in order for my fitness function to be guaranteed to be non-negative, I need this capital M, this new hyperparameter, which shifts that negative up to guarantee that this difference here is going to be positive. So that is one simple common way to generate fitness functions if you're doing a minimization problem, is by figuring out for your feasible set how, what are the bounds on your optimization objective. Create a constant that is larger than those bounds so that when you do m minus that, it will never be negative. That is one way to do that. Now that I have that hyperparameter, then the question is, what else does that hyperparameter do? So one of the things it does is make sure that f is not negative. The other thing that it does is affects the selective pressure. Let's say that I knew that my little f, the highest it could get was 10. So if this little f here, if the highest it could get was 10, and I wanted to minimize little f, then I just need to choose a big M that is 10 or higher. So if I choose it to be exactly 10, then in the worst case scenario, my fitness is 0. In the best case scenario, my fitness is 10. Or whatever, I don't know what the other bound is on the other side of this little f. But point is, if the biggest this can get is 10, I can set this equal to 10. But why 10? Why don't I set this equal to 100 or 1,000? So I guess I could write this down here. So as an example, if f is less than or equal to 10, then m could be 10, or it could be 100, could be 1,000, and so on. So can anybody think, what do you think is the impact of choosing an m that is far larger than it needs to be to make sure that the fitness function is non-negative? How does that affect how different individuals compare to each other? What was that?
Well, everything just looks the same.
Looks the same. What do you mean by that? They look the same.
It'd be hard to have something that really differentiates or selects based on fitness.
Yeah, so it'd be hard to tell the difference between genotypes, because let's say you've got, normally you've got genotypes that are, say, between 0 and 10. So in that case, it might be that one genotype is like the one genotype's down, so I'm trying to minimize errors. Let's call F as like errors. And one of them, like, it's like that kid behind the counter at Paris Baguette who was just really, was sort of the paperweight employee. He's just constantly making errors. And so F is equal to 10. But the manager who's walking around doing everything right, she's making no errors. So her F is equal to 0. So I can, on that scale from 0 to 10, there's a huge difference between those. So if my fitness function sets n equal to 10, then the manager is going to have a fitness of 10, and the employee, sort of as our baseline for failures, has the fitness of 0, and there's a huge difference between them. Now, if I instead set my m constant here, this affine constant, this shift, to 1,000, then now the difference between the two of them is the difference between 1,000 and 990. And so in that case, I don't see as much of a fractional difference between them. So if my selection operator is looking for relative fitness differences, the relative differences, so I can mention this here, relative fitness differences will be lower. And that adjusts selective pressure. So this is a hyperparameter. So just mention here, this is a hyperparameter. And the idea here is that hyperparameter, if you notice that your genetic algorithm is converging very quickly, it's converging to local optima, like it says like, okay, I get that that's a good position, but I'm pretty sure there's another one elsewhere. you could then increase that M to something very large, and that would mean that it's going to be less sensitive to small differences in fitness, and it'll leave those in the system so that they're free to explore. It maintains diversity in the solutions for longer until there is a huge difference in fitness. So with a small M, then you're very sensitive to small differences in fitness. But with a large M, you only start killing off individuals if there's a huge difference in fitness between those individuals. So we now have one lever to adjust selective pressure. And this will make more sense as we talk about selection operators, but I'm trying to define fitness first. Questions about that? I can probably move ahead because it's going to take 30 seconds for it to snap in there. So another example fitness function that gets closer to that neural network example is that if I take this and I imagine, let's say I want to minimize the loss of a simple neural network that's implementing this kind of linear regression or solver solution here. So this is kind of this middle term, this AX minus B, is sort of saying if I've got a genotype that's implementing X, then what I'm trying to do here is to minimize the difference between A, which might be a set of data. It's like I took a bunch of samples and I have different rows. So I'm sampling different things and I have different independent rows. So that fits into a matrix. And so, so I've got some matrix A, which is a part of my problem. I'm varying X and I'm trying to make AX equal to B. And so I'm trying to minimize this thing. But I also I add this regularizer penalty. And so I've got this thing doesn't depend on A, it only depends on my genotype. And so effectively saying I want the size of my genotype to be small. I'm penalizing large, and I'm not saying large in terms of number of genes, but large in terms of the actual alleles that are there. So let me flip back there. So just in a second, hopefully that comes back there. But what I'm looking at here, which I think should pop up just a second. We're seeing history of what it just looked like a second ago on the tablet here. Okay, so the idea here is this is one way that you can create multiple objective, how you can we call this scalarization of a multi-objective problem. This is the poor man's solution to a multi-objective problem. This is not how we're going to do multi-objective in Unit 3. But this is how you start. When you first start thinking about multi-objective, this is the first thing that comes to mind. And this is what most of the machine learning community does when they do multi-objective. You take your two objectives and you add them together and you put a weight on one of them, which represents the trade-off that you desire between them. So that lambda between there is a hyperparameter which says how important this second objective is, the regularizer objective, relative to the first objective. And so if you make this lambda very large, then it's just going to create, it's just going to select genotypes that have a very small 2 norm, their squared size is very close to 0. If you choose a lambda close to 0, then it's going to focus on solving this problem, bringing AX close to B. So, and so when you blend those, then you end up getting solutions that do a pretty good job at both. And so they represent a trade-off that is captured by this lambda here. And so we can take that same loss function that you might have in a neural network, which would be this plus lambda that, And we can just do, just basically subtract the whole thing. So that minus distributes through, and that gives us this fitness function. And so that is another example of a fitness function. And then likewise, playing with that M plays with selective pressure. So it's basically the same example we had before, but I'm just trying to show you that now we theoretically can talk about two different characters, this character and that character, or this trait and that trait, and how they combine. Now, it's a little bit semantics at this point, though, because you might say, well, no, this whole combination is a character. And so this is really just one single character, and because my genetic algorithm doesn't care that I'm viewing the genotype in two different ways, that way and combining them together. My genetic algorithm, for all it just cares that I have one way to view the genotype. So it's not really two characters. It's kind of cheating to call it two characters because we're doing this scalarization. In Unit 3, we will define a completely different way to sort genotypes that does consider these two characters separately and creates a way to sort them without having to introduce these stupid lambdas. Okay.
So that's just another hyperparameter that we're adding.
Yeah, right. And this is a hyperparameter that you'd have, like if you were anybody who's had to train a neural network, these regularization things, like this is almost like this is like a hyperparameter introduced. This isn't a GA's hyperparameter. This is like a hyperparameter of who set up the problem. So somebody who set up the problem has said that this is how I'm penalizing these types of solutions. So this matters to me in a way that's sort of more central to my problem. Whereas this hyperparameter M actually affects the function of the GA. In the shaping the dynamics of the GA, lambda is communicating the stakeholder's preference for having these compact solutions versus solutions that solve this AX equal B problem. So they're both hyperparameters, but this is the GA specific one is the one outside here. This one is sort of like, again, I really could just bundle this whole thing. This really is just one, separate, F loss function, and I could just forget whatever's inside it, and it would be just fine. It's just a black box. It might as well be a simulation or something. Questions? Does that make sense? Okay, and then another example fitness function, which gets us to this idea of fitness scaling, is not everything has to take this form, where it's M minus the loss function. An alternative way that is somewhat elegant is to use a sigmoidal function like this one that just popped up here. So in this case, then the idea here is I have my character, but I'm going to scale it with this. I've got my character on this horizontal axis down here, the bottom, and I'm then mapping it to this scaled fitness function on the y-axis here. And the way this works is that if you have very large values of the character, so if the character goes to positive infinity, so the f of x, if f of x goes to positive infinity, then big F of x, the fitness function, will go to 1. And if little f of x goes to negative infinity, then big F of x will go to 0. So it's just your standard S-shaped, sigmoidal fitness function here, or a function here, which has a bunch of nice features. For one, maximizing fitness also maximizes little f, which is fine. It's going to die again. So I'll erase a couple of things while I let it reconnect. Sorry about this. I thought not using the room computer would resolve some of these issues, which it has, but something else more fundamentally wrong about this room. So I might end up just having to tether to the HDMI and stand over here in the future. Not screen sharing yet. Okay, there we go. Start broadcast. All right, so coming up, I'll just go ahead and erase these. There's no time for drama. And the idea here is that big F, is already meets our non-negative constraint. So we don't have to add some arbitrary shift, regardless of what the character values are. The character values can be negative, they can be positive, but this ensures that it's automatically non-negative. In fact, the fitness is just bounded between 0 and 1. That might be kind of nice or convenient. And there's this hyperparameter, capital T, which I've put up here, e to the capital T, which is this kind of, I'm going to call it a half saturation constant. And so this hyperparameter determines the value of the character where the fitness is right there in the middle of this sigmoidal rise, at this inflection point. Now notice that at that point, that's when fitness values are most linear. So there we go. So around that point at 0.5, that's when small changes in your character value lead to similarly scaled changes in your fitness function. But out for very large values of the character, then you don't see very much change at all in the fitness function. And for very small values of the character, you don't see very big changes in the fitness function. And so in a second here, that loop there on the right should change to a loop down here on the left. All I was doing was circling down here. So large values of the character give you no changes in fitness. Small values of character give you no change in fitness, but values around this particular point, so character values around T, then you actually see that difference there. So this idea of fitness scaling allows you to sort of focus on things that are smaller than T, so F values that are smaller than T, you don't really care about. You're sort of things that are much smaller than T or much larger than T, you don't really care about evolving the system very much. You're sort of preserving diversity at small values of the F function and at very, very large values of the F function. And but then getting your higher selection pressure for these intermediate values. So that might make more sense for your particular character that you're looking at there. You might say that once the character is really, really large, I don't care about differences between two really, really large values of that character. Like at that point, they're all about the same to me. Or once character values are really, really small, those things are pretty much the same to me. But when the character values are in this range, I might just expect that normal character values are going to be in this range, and it's weird for them to be large values or something like that. Then this works out really well. So this is an example where it also just generally compresses the difference. So you might have F values. These things might vary across all of the genotypes you could consider. This F value might go from -1 million to +1 million. So simultaneously, you could have a genotype that's scoring -1 million and another genotype in the same population that's scoring, almost +1 million. And if you just use those F values as your fitness values, you would not preserve any diversity in that system. You'd lose all of those -1 million ones. But by doing this, you greatly reduce selective pressure, and you make it more likely that unless the character values are really, really different, you're always going to maintain some diversity. So it just sort of settles things down. So that's an example of what we mean by fitness scaling. So I defined this as a fitness function, but as we'll see, you can also define this as a scaled fitness function where actually this little f, you can, in fitness scaling, so I'll say this here, can be a lower level fitness function. So what I'm writing right now, which hopefully should come up soon, is that this little f here itself can be a lower level fitness function. So what we'll see in the algorithm of the GA is that sometimes it's convenient in the GA, and commercial GAs like the ones you see already implemented in MATLAB, for example, they will let you define or structure a base fitness function and then compose it with a fitness scaling function. And you can think of this as one of those examples of that fitness scaling function, where you might establish fitness that's maybe it's non-negative, but it just happens to have a very large scale. And then you say, but then once I get my fitness values, once you calculate all those fitness values from all those traits, then take that and scale it like this so that I bring all my fitness values into this close range to reduce the selective pressure. So it's either a fitness function or a composite fitness function. Questions about that? Does that make sense? So just different ways to enforce non-negativity and change the scale, because the scale is where selective pressure lives, where our bias on selective pressure lives. It's how we tune selective pressures, through tuning these scales.
This is just logistic regression, right?
This is a function that is used in logistic regression. I'm not doing logistic regression. So I can use a logistic as my sigmoid. I could use lots of other things as my sigmoid. I could use a hill function as my sigmoid. So you can come up, if you like a sigmoidal scaling function, I can use a hyperbolic tangent as my scale. So there's a bunch of different sigmoidal functions that if you like this feature, you might like to reshape like where it goes to zero, and there might be nuances of how to change the shape of this S-shaped function, and you can choose different ones there. So because I'm using something that looks like a logistic, I'm not implying though that this is a logistic regression problem, but it happens to be using the same sigmoid use in a logistic regression problem. But just like choosing activation functions in a neural network, you can choose all sorts of different types of activation functions. You can choose all sorts of different types of scaling functions. Okay.
So this would also have things like the rectified linear unit, ReLU, and software.
Well, I don't. So there are. So we haven't got the neural networks yet, but in neural networks, there are a bunch of different activation functions like ReLU, so rectified linear units, and so on. Those, you can think of those as trying to scale the outputs of neurons. And so in some ways, mathematically, they are in the same neighborhood as what we're talking about here. So it shouldn't be a surprise that some of the scaling functions that you can use for fitness scaling might actually look exactly like activation functions you'd use in a neural network. I would claim that that's because the neural network folks are using a design paradigm that existed before them when they came up with those activation functions. And we're tapping into that same design paradigm here. All right, so it's bringing up broadcast again. Again, apologies for this cutting out. Tried to avoid having to tether myself by the lectern, but it may be that this room just requires that. All right, so with that, maybe I'm just going to start erasing things here. Okay, so this is just summarizing what we mean by fitness scaling. That's the point of fitness scaling. Ensure fitness is non-negative and adjust selective pressure. All right, so now let's start then laying out the algorithm structure so that we can then start filling in all these operators, maybe a little bit today, but then definitely on Thursday. So the basic genetic algorithm structure is you start with a random set of genotypes, so random initial population. And from that random initial population, you just draw. It could be completely random. Every gene, you could go 4i and 1 to the number of individuals. So for every individual and across every gene, you can just draw a random allele. And then that will give you a random population to start with. That is a perfectly reasonable way to start your genetic algorithm. A lot of commercial GAs will either do that for you or they'll ask you, do you want to give us the initial population? Because you might have kind of almost like Bayesian optimization. You might have some prior information that this region of the decision space is where we're trying to really start exploring. So you might start, however it is, you start with some initial population, and then you calculate all the trait values. Another way to say that is you calculate the optimization objectives. So you would tell your MATLAB, tell your PyMu, or whatever framework you're using, what optimization objectives you have. And then those optimization objectives for every individual get calculated, and those get turned into these trait values. For a single objective problem, like we're going to start out with a GA, it's just going to be one trait value for one character, one optimization objective. After you've calculated that, then you, and some of the ordering of a couple of these things could be flipped, so it's a little bit arbitrary, but you can check for your termination conditions. So termination conditions, they could be the number of generations, how many times have we gone through this loop. They could be convergence. So are the trait values that I see, like if I could look at the span, or I could look at what is the maximum difference between the trait values in this population. What was it in the previous population? So if it looks like my population's trait values are really close together, so that like really I'm not like exploring the space very much, that might be, I might say once those are so small that, that I'm not anticipating much more movement, I might end the algorithm. So you can come up with these convergence conditions or general termination conditions, and generally speaking, a commercial optimization algorithm will give you a bunch of different options there that you can set. Once you decided you're not going to terminate, then you go and calculate your scaled fitness values. So you know your characters, and so the idea here is that you then turn those characters into these non-negative fitness values that allow you to sort these individuals from high quality to low quality. Once you have these scaled fitness values calculated, then how does this, we now get to like where the sorting action actually has consequences. And that's where, and so we have to define a selection operator, which will be the next thing that we'll formally define. We select parents. That's what that says. Once I get rid of these, then these will be more readable. And so the idea there is that I'm selecting high fitness individuals that will be the source of the newest generation. So I've got a generation of individuals. Some individuals have done very well. Other individuals aren't doing that great. We're assuming that the individuals that don't do that great do not survive to reproduce. So by selecting parents, I'm basically selecting the ones most likely to reproduce. And that we'll do through a so-called selection operator, which we'll get to in just a second. And then we'll continue on Thursday. So it's not going to be a mad dash or anything. So then after I have my parents, now I use those parents to generate new offspring. So these offspring will vary, and they will be similar to the parents, but then they will also vary off of them in ways that we get to choose how they vary. So these parents then end up help us generate new variation, new offspring. And those offspring that are based on the parents then get mutated in a way that is not based on the parents. So they get independent mutations added to them. And then that gives us our new population. So it's a population of individuals who include parents, possibly elites, which are like high fitness individuals we never want to throw away until another individual shows even higher fitness than them. And then offspring, which are based on those parents, but differ in ways that we can define. And then mutations, so variations of those that are not based on the previous parents, that are just independent variation added into the population. And then that generates a new population for us to then start this off again. So in each one of these cases, I've defined a bunch of things that we have control over or the person running the algorithm has control over. I mentioned the termination conditions. When we check for termination, that is a degree of freedom in the algorithm. I've mentioned the scaling functions. You can choose different scaling functions or different parameters of existing scaling functions. I've mentioned that when you select the parents, erase all these, so that eventually it'll pop up here, how you go from a population's individuals to those that have been given the opportunity to reproduce. That is a so-called selection operator. And that selection operator, which we're going to choose, will operate probabilistically. So higher fitness individuals will be more likely to be selected. But a low fitness individual might still get selected just by random chance. That's where drift comes into play. So the way drift acts on a genetic algorithm is in the selection operator when things go kind of counter to what fitness might predict. So you might have a bunch of high fitness solutions, and yet by random bad luck, the selection operator, which sets up the probability function, which draws individuals into the parenting pool, it will likely pick high fitness individuals. But sometimes high fitness individuals will have bad luck and they won't be selected. Sometimes low fitness individuals will have good luck and be selected. And that is modeling the output of genetic drift. And then when we then go from parents to offspring, then there is something that was called a crossover operator that we have to implement. We're going to get to there, and that's going to have its own hyperparameters. And like we'll see, that models the function of sex in biological systems. So sex is interesting in that it does not change the distribution of alleles immediately, but it changes how they assort, and that will allow fitness to act on them differently. So some alleles might have always been piggybacking on other alleles for other genes that were really high fitness. Now, once you separate them, then suddenly it can't ride on the coattails of that other gene. And then fitness, the high fitness that was coming from one gene was creating a shadowing effect, preventing the other gene's deleterious effects from being selected out. Sex allows them to separate from each other so that now you can get the gene with the high fitness allele now gets to try out another gene next to it with a different allele that might be better off than the one that used to be coupled together. So that's what we'll see in the crossover operator. And then mutation, we're going to have a parameter based on the mutation rate, and that's what mutates this new population. So all of those, and I also mentioned the initial population, key parameters are the size of the population and the genetic architecture, or at least the number of genes. So all of these things across the bottom, the population size and all of these things here, these are the hyperparameters of a genetic algorithm. And those are the things that's why we went through all of the kind of population and quantitative genetics earlier in this unit, is that this is a complex system. It's a complex optimization approach. And it's hard to build, at least initially, kind of an intuition about how these different parameters trade from each other. But it's my claim that what the population geneticists and quantitative geneticists have given us as conceptual tools that help us figure out that if we're seeing our GA just isn't quite cranking out the solutions that we think it should, then we have ways that use that intuition from biology to adjust these parameters so that we can get a better operating GA. Okay. All right, I think this is probably a good place to stop. Any questions? So what we're going to do next time is we're going to now start introducing these operators. So what does a selection operator look like? There are three main selection operators. What does crossover look like? What does mutation look like? And those are sort of the big things, how these things work together. About any GA looks roughly like this. GAs differ in this and whether they have alleles. Question?
Where does what's that word called? Selective.
Pressure, coefficients.
Natural selection come in the number of reproductive offspring.
Well, this here is particularly the portion here. Whenever I say select, that's kind of, that is shorthand for natural selection. So it's theoretically this is artificial selection, but the idea here is that that we're imagining that nature is sort of like the optimization objectives that are getting calculated and that are pushing these things around. So whenever we say selection, that we're mimicking natural selection. So we're not mimicking sexual selection. Sexual selection would be like if one individual had a preference for another individual and that affected who rejoined here. But and you can theoretically add that to operators, but this whole process here is the process of natural selection, or you could think of it as breeding. So it could also be viewed as artificial selection. So it's like we've got one genotype and it's got some traits we like, and we then try to breed more genotypes that have traits that are like that. All right, well, anyways, there are people at the gate, so if you want to have other questions, feel free to come up. Otherwise, we're going to pick up and start defining the shape of the operators that go into a genetic algorithm.